And now it's time to start our video on enzymes. So just as a brief note, if I forget to say it later, there are already videos posted to this channel about specific mechanisms of proteases. We use chymotrypsin as an example of a protease, something that breaks down protein mechanism. We also have videos on the Michaelis-Menten and the Lineweaver-Burke graphs, how we derive them mathematically, where we get them from, what we can use them for, the different types of inhibition. So we will not specifically cover them during this section, but those videos are already posted. So let's get into enzymes. And so we're going to start with a little bit of the history of discovery, just because it's very interesting. Change to black, because it's very interesting to see the thought that went in to how we discovered enzymes. And so the first person who really looked at it was a guy by the name of Louis Pasteur. Louis Pasteur was interested in fermentation and vitalism, right? This is in the 1850s. And so what he's arguing is that you need living cells for fermentation to occur. Obviously, he was big on pasteurizing, killing those living things that spoil food. And so what he said is you needed the living cell. The living cell was the important part of the function of what it did, right? And so you needed it. That's vitalism, life. And then we get Buchner coming in the 1890s. And he showed that living cells were not required, and in the absence of the living cells, the machines inside of them still worked. And so there is something inside the cell that does a function. And so, unfortunately, Buchner died during World War I, and so he wasn't able to expound even further on what he said. And then, the next step was Sumner in the 1920s. He crystallized urease. And so he made a crystal of urease, and what he said was that crystal was entirely protein. That's what made up that crystal was protein. Now this was a very controversial statement at the time, because again, we really didn't know what things made up protein, really, what made up enzymes themselves. And then we get a guy by the name of J.B.S. Haldane. He was working in the 30s, so another decade has gone by. And he actually, what's funny is that he actually worked in population genetics. So he wasn't a biochemist. But what he proposed was that that an enzyme has multiple weak binding interactions that helps decrease the free energy, or gives enough free energy for the catalysis, right? He had no idea how this worked. He just proposed that, through multiple binding interactions, you would get stabilization of the transition state, and then you would be able to get catalysis. So, then there's a couple guys named Michaelis and Menten who came along and proposed a model for how that might look. And so their model looked like this. And it's a very well-known model. And so we have a little bit of constants there. And so, just a couple things. This K3 can also be known as Kcat, as the catalytic step, right? We're turning substrate into product. Notice, substrate into product is not reversible. Once an enzyme turns a substrate into product, usually it cannot be turned back into substrate. We draw these two as an equilibrium, but they're really not. Usually what ends up happening is, and I can draw them to scale using this red line, is it's like this. That usually once an enzyme binds to its substrate, it is pretty much going to go to catalysis. However, there can be some dissociation. So Michaelis-Menten proposed this model. We know this is a lock and key model. We'll talk a little bit about what that means, or it's the start of the process of thinking as enzymes as locks and keys. And then another pair, Briggs and Haldane, came along and slightly modified the Michaelis-Menten equation. And so what they said was, here's our enzyme. Yeah, let's do them both in black. Here's our substrate. Those two will interact, and we'll get an enzyme substrate complex. And then what happens is there's a slight modification, and we get an enzyme product complex, and then we get dissociation, which actually should just be drawn like this, where the enzyme gets regenerated and the product is made, and so they're released. They came along and they said, well, when an enzyme binds to its substrate, it's really a strong interaction. It's like a lock and a key, right? There's a specific substrate for a specific enzyme active site, and that is true. But what happens is it's not exactly like what we usually think of it as with lock and key. And so what would happen if it was lock and key, right? So that it's a very specific interaction when the substrate binds the enzyme. Here's what the free energy diagram would look like. Free energy reaction process. Okay, you'd have your substrate here, it would bind to your enzyme, and then it would be made into product. Okay, so this is obviously not drawn to scale, but what happens is this enzyme substrate complex right here is of a lower energy than just the substrate plus the enzyme. Right? And so it would be very stable in this portion, and so that this energy that would need to be overcome, this activation energy, would just be too high for it to actually act as a catalyst. Now, this does occur, right? So just because we say lock and key does not occur for enzymes, or at least it's not the prevailing view, does not mean that it doesn't occur anywhere with any proteins. An antibody-antigen binding does do this, right? To where the antibody binds to its antigen, it is more stable than the two of them just freely floating around. So then what actually happens, I'll draw another one of these. Oops. Free energy, reaction progress. And so normally we have this. But now what we're going to talk about is an induced fit. And that induced fit, I guess I could just draw it all, drops it down to there, right? So it is catalyzed. And the reason why is there's multiple weak binding interactions, like Haldane said, to help stabilize that form. And so what we can think of, and it's really hard to see because you can't see me, is that enzymes breathe. And so they're kind of like a glove, right? Hand in glove. So the glove is very kind of floppy in the absence of any hand, but yet it still has its shape. Once you put your hand in it, then it is now more structured. And so the way we can think about these enzymes, it's kind of like this. So there's our enzyme. Here's our substrate. Those two are going to bind. Like that. And so within this region here, there are a bunch of hydrogen bonds, a bunch of salt linkages, just a variety of different interactions that can help stabilize this form and lower the activation energy. And then what happens is there's the transition state. I know my enzyme keeps changing. Oops, that's not what I want. And so now we make product, and then what happens is the product gets released, the enzyme gets regenerated, and so that is how we start to think about proteins and how proteins act. Some proteins have coenzymes. Right? And so coenzymes are just non-protein compounds that are necessary for the function of the enzyme. Something like NADH or NAD plus that gets turned into NADH. There's a variety of different types of coenzymes that need to be. There's also cofactors. So usually the difference between a coenzyme and a cofactor is one is organic, the other one isn't. Usually when we think of coenzymes, we think of organic molecules, something like NAD+. Usually when we think of cofactors, we think of a metal, something like zinc or magnesium or calcium. And so what we can have are a protein, let's say horse liver alcohol dehydrogenase. As by itself, it is not active. It is known as an apoenzyme. Right? An apoenzyme is not active. We can add its cofactor, zinc, to it. And what we get is horse liver alcohol dehydrogenase associated with the zinc. Now it can function. Now it can do what it needs to do. This is known as a holoenzyme. So that is an enzyme that can function. That is functional. HLADs are very common generic enzymes. Every mammal has one in their liver. And so what happens is we can do a couple different types of graphs. Let's look at activity of an enzyme versus pH. And what we usually see is this nice bell curve where 7 is or so is in the middle. It's maybe offset to the right a little bit. Digestive enzymes are found down here, active at pH of 1. We can do another graph. And so this is very common in a lot of the enzymes where activity and temperature. And we see it where at 37 degrees or body temperature, the majority of the enzymes are active. Whereas if you're at this end, if you're lower than 37, they're slowing down and eventually will denature even at cold temperatures. And if you heat them up too much, then they will also be denatured, like cooking eggs in a skillet. So, what are some different types of enzymes? So these are just some generic types of enzymes. One thing to notice is all enzymes end in ASE. So urease is an enzyme. Usually what happens is you look at what's in front of the ASE and it will tell you what its substrate is, or at least what chemistry that it does. So alcohol or horse liver alcohol dehydrogenase, can do this. Turn ethanol with the help of NAD, right? And it needs zinc to function. Can turn it into this. Right? Turns it into acetaldehyde. And so what this is oxidoreductase. Right? So an oxidoreductase, there's an oxidation and a reduction reaction that occurs. Usually if you see an oxidoreductase, it has to have something like this. That's a very common thing. It's a proton carrier. We'll talk about it hopefully later in another video. And so horse liver alcohol dehydrogenase does this. Dehydrogenase, if you see a dehydrogenase, you're going to remove a hydrogen, and it will be an oxidoreductase. Here's another one. Let's say we have DNA 1 plus another strand of DNA 2, and there are enzymes in our body that can put them together, and these are known as ligases. Right? So this would be a DNA ligase. A ligase places two large polymers together. The next one that we'll see later in glycolysis and the glycolysis videos are already posted if you'd like to take a look and see where this occurs actually in glycolysis, is this. We have, I'm going to be lazy and just draw the phosphate like that. We have DHAP, a molecule called DHAP, or dihydroxyacetone phosphate. There is an isomerase, specifically the triose phosphate isomerase, but isomerase can then turn it into this. It performs an isomerization reaction, right? So just changing how the molecules are bound, the elements are bound, and you get glyceraldehyde 3-phosphate. Right? So this occurs in glycolysis. So isomerases just change the structure and not anything else, and how those atoms are bonded. We also have this. So in the citric acid cycle, we're going to see this. Run out of room at the bottom. So this is fumarate. We can break that double bond by adding water to make this. To make malate. This enzyme is known as fumarase, but the classification of it is a lyase. Right? A lyase is you're adding something to something else to break the double bond. And then the last one, I'm just gonna write it out. I'm not gonna really show you the chemistry, but it's a transferase. Right? You're transferring one one group to another group. Usually they can be pretty large, but so that's transferase. So those are just the different overall views, or the overall different characterizations of these enzymes. In the next video, we will talk about a little bit about what can the Michaelis-Menten graph actually tell us. Right, so a little bit more extra information and maybe a reiteration of some of the stuff in the video about it. And we're also going to talk about enzyme regulation, not necessarily the Lineweaver-Burke stuff, but just in the cell, how are these enzymes regulated?